Good morning, class.
Oh man, Professor Scott. Oh my gosh, my eyes have been like really sensitive to light lately. Can you explain why this happens?
Well, it's actually funny you say that because today we're going to be talking about the eye and how light is transduced into the eye and how we process information in the brain. To have a better way of looking at it, we can look at the camera.
Okay.
As we look into the camera, we can see that the iris would be the shutters of the camera, and the pupil would be the lens as it dilates and constricts to certain light. Light rays enter the eye through the cornea, the clear front window of the eye. The cornea's refractive power bends the light rays in such a way that they pass freely through the pupil, the opening in the center of the iris through which light enters the eye. Rods have one type of visual pigment, while cones have three different pigments that are closely related to rhodopsin. The visual pigments of cones are excited by different wavelengths of light, allowing us to see in color. White light is a combination of colors, as demonstrated when you separate white light by passing it through a prism. The eye contains cones for red, green, and blue light. Each cone type is stimulated by a range of light wavelengths, but is more sensitive to a particular wavelength. The human eye functions over a 100,000-fold range of light intensity. Most of this ability comes from the sensitivity of the photoreceptors, but the pupils assist by regulating the amount of light that falls on the retina. In bright sunlight, the pupils narrow to about 1.5 millimeters in diameter, when a parasympathetic pathway constricts the circular pupillary muscles. In the dark, the opening of the pupil dilates to 8 millimeters, a 28-fold increase in pupil area.
Professor Scott, so I heard you mention something about photoreceptors. Can you explain that a little bit more deeper?
Yes, we can go into that right now. There are two types of photoreceptors that react in bright light and low light. Rods function well in low light and are used in night vision when objects are seen in black and white rather than in color. They outnumber cones by 20-to-1 ratio, except in the fovea, which contains only cones. Cones are responsible for high acuity vision and color vision during the daytime when light levels are higher. Acuity means keenness and is derived from the Latin word acure, meaning to sharpen. The fovea, which is the region of the sharpest vision, has a very high density of cones. Hyperpolarization by light is caused by the shutting off of a flow of ions. In darkness, part of the receptor membrane is more permeable than the rest of the membrane to sodium ions. Consequently, sodium ions continually flow into the cell layer, potassium ions flow out elsewhere. What causes this biochemical gradient is the protein part of the opsin changing shape.
Professor Scott, I heard you mention something about opsins. So can you dig a little deeper with opsins for me and just tell me what significance they play in our eyesight?
Opsins are a group of light-sensitive proteins found in photoreceptors. There are 5 groups involved in vision, mediating the conversion of a photon of light into electrical chemical signal. After passing through the opening of the pupil, light strikes the lens, which has 2 convex surfaces. The cornea and lens together bend the incoming light rays so that they focus on the retina, the light-sensitive lining of the eye that contains the photoreceptors. When viewed through the pupil, the retina is seen to be crisscrossed with small arteries and veins that radiate out from one spot of the optic disc. The optic disc is the location where neurons of the visual pathway from the optic nerve exit the eye. Neural pathways for the eyes are shown above. The optic nerves from the eyes go to the optic chiasm in the brain, where some of the fibers cross to the opposite side. After synapsing with the lateral geniculate body of the thalamus, the vision neurons of the tract terminate in the occipital lobe of the visual cortex. Collateral pathways go from the thalamus to the midbrain, where they synapse with efferent neurons of cranial nerve 3 that control the diameter of the pupils. So from everything that we've talked about today, what do you think causes our blind spots?
After everything that we've talked about, I've come to the conclusion that because of the optic disc not having any photoreceptors, I believe that the images cannot be processed in this region, which gives them the name blind spots.
That is correct. So to sum up everything that we've talked about today, vision is the process of which light is reflected from objects in our environment into a mental image. And I've drawn a flowchart to better understand how images are processed into our brain. So light enters the eye, and the lens focuses the light on the retina. Photoreceptors of the retina transduce the light energy into an electrical signal. And the electrical signal transfers this into neural pathways from the retina to the brain during processes electrical signals into a visual image. So I hope everyone
everyone has learned a lot from today.